Genetic engineering is not a new concept. For millennia, humans have been manipulating genes without even realizing it. They selectively bred plants and animals, favoring desirable traits. This led to the development of crops with higher yields and animals with specific characteristics. However, these early attempts were based on trial and error, lacking the precision of modern techniques. The discovery of DNA structure in the 1950s revolutionized our understanding of heredity. It paved the way for genetic engineering as we know it. Scientists began experimenting with directly modifying genes in the latter half of the 20th century. Early techniques were crude, often involving the use of radiation to induce random mutations. These methods were like shooting in the dark, hoping for a lucky hit. While they advanced our understanding of genetics, they lacked the precision and efficiency needed for targeted gene editing. The real breakthrough came with the development of recombinant DNA technology in the 1970s. This technology allowed scientists to insert specific genes from one organism into another. This opened up a world of possibilities, from producing human insulin in bacteria to developing genetically modified crops resistant to pests. These early successes fueled further research and development in genetic engineering. However, the techniques remained complex, time-consuming, and expensive, limiting their widespread application. The field was ripe for a revolution. The revolution arrived in the form of CRISPR-Cas9, a revolutionary gene editing tool. CRISPR, short for clustered regularly interspaced short palindromic repeats, is an ingenious defense mechanism found in bacteria. It acts as a bacterial immune system, protecting them from invading viruses. When a virus attacks a bacterium, the bacterium captures snippets of the viral DNA and integrates them into its own genome. These viral DNA fragments are stored in a specific region of the bacterial DNA known as CRISPR. The CRISPR region acts like a genetic library, storing information about past viral encounters. If the same virus attacks again, the bacterium can quickly recognize and neutralize it. This recognition is mediated by short RNA molecules transcribed from the CRISPR region. These RNA molecules, known as guide RNAs, bind to the matching viral DNA sequence. The guide RNA then directs a special enzyme called Cas9 to the target DNA. Cas9 acts like a pair of molecular scissors, cutting the viral DNA at the precise location specified by the guide RNA. This cut disables the virus, preventing it from replicating and causing further harm. What makes CRISPR so revolutionary is its adaptability as a gene editing tool. Scientists realized that by providing a custom-designed guide RNA, they could direct Cas9 to cut any DNA sequence, not just viral DNA. The advent of CRISPR-Cas9 has been nothing short of a paradigm shift in genetic engineering. Its simplicity, precision, and affordability have democratized gene editing, making it accessible to a wider range of researchers. This has led to an explosion of research in various fields, with medicine at the forefront. CRISPR holds immense promise for curing genetic diseases. Thousands of diseases are caused by errors in a single gene. CRISPR offers a way to correct these errors, potentially curing these diseases at their root. One of the most notable successes of CRISPR has been in the fight against HIV. In 2015, scientists successfully used CRISPR to cut the HIV virus out of human cells in a lab setting. This groundbreaking work raised hopes for a cure for HIV, a virus that has plagued humanity for decades. Subsequent research in animal models showed even more promise. In a study conducted on rats infected with HIV, CRISPR treatment was able to remove more than 50% of the virus from the animals' bodies. While these results are still preliminary, they highlight the potential of CRISPR to cure not only HIV, but also other retroviruses that insert their genetic material into the host's DNA. Beyond HIV, CRISPR is being investigated for its potential to cure a wide range of other diseases, including cancer, genetic disorders like cystic fibrosis and sickle cell anemia, and even neurodegenerative diseases like Huntington's disease. Aging, once considered an inevitable part of life, is now being viewed as a disease that can potentially be treated or even reversed using gene editing. CRISPR has emerged as a powerful tool in the quest for longevity, offering tantalizing possibilities for extending human lifespan. While the idea of significantly extending lifespan was once relegated to the realm of science fiction, CRISPR has brought it closer to reality. Scientists are exploring various ways in which CRISPR could be used to combat aging. One approach focuses on targeting the hallmarks of aging, such as cellular senescence, telomere shortening, and genomic instability. By repairing damaged DNA, removing senescent cells, and rejuvenating tissues, CRISPR could potentially slow down or even reverse the aging process. Another promising area of research is the manipulation of genes that influence lifespan. Studies in model organisms have identified several genes that play a role in determining lifespan. By tweaking these genes using CRISPR, scientists have been able to significantly extend the lifespan of worms, flies, and even mice. While these results are still in the early stages, they offer a glimpse into the possibility of manipulating human lifespan using gene editing. The prospect of extending human lifespan raises profound ethical and societal questions. Section 5. The ethical tightrope of germline editing. Germline editing, the modification of genes in reproductive cells like sperm, eggs, or embryos, presents a profound ethical dilemma. Unlike somatic cell editing, which affects only the individual being treated, changes made to the germline are passed down to future generations. This raises concerns about the potential for unintended consequences that could affect the entire human species. The possibility of germline editing became a reality with the advent of CRISPR. In 2015, Chinese researchers reported the first attempt to edit the genomes of human embryos. While the study used non-viable embryos and was not intended to establish a pregnancy, it sparked a global debate about the ethics of germline editing. The scientific community largely condemned the experiment, calling for a moratorium on germline editing in humans. The main ethical concerns center around the idea of altering the human gene pool without fully understanding the long-term consequences. Critics argue that germline editing could lead to a new form of eugenics, where parents select desirable traits for their children, potentially exacerbating social inequalities. Others worry about the potential for unintended consequences, such as off-target edits or unforeseen interactions between genes that could have devastating effects on future generations. The debate over germline editing is complex and multifaceted. Section 6. Designer babies, a slippery slope. The concept of designer babies, children whose genetic makeup has been artificially selected to enhance certain traits, has long been a staple of science fiction. However, with the advent of powerful gene editing tools like CRISPR, the possibility of designer babies is moving from the realm of fiction to a potential reality. This raises profound ethical and societal questions about the limits of genetic manipulation and the potential for exacerbating existing inequalities. The ability to select for specific traits, such as intelligence, athletic ability, or physical appearance, could lead to a future where parents with access to these technologies have an unfair advantage in shaping their children's destinies. This could create a genetic divide between the haves and the have-nots, further entrenching existing social hierarchies. Moreover, the very notion of desirable traits is subjective and culturally influenced. What one society deems desirable may be considered undesirable or even abhorrent by another. The pursuit of genetic perfection could lead to a narrowing of human diversity and a devaluation of individuals who do not conform to these artificial standards. Beyond the ethical concerns, the scientific feasibility of designing complex traits is still debatable. Section 7. Unforeseen consequences, navigating the unknown. While the potential benefits of CRISPR are undeniable, it is crucial to acknowledge and address the potential risks and unintended consequences associated with this powerful technology. One of the primary concerns is the possibility of off-target edits. CRISPR is not perfect, and there is a risk that the Cas9 enzyme might cut DNA at unintended locations, potentially leading to harmful mutations. While researchers are constantly working to improve the precision of CRISPR, the risk of off-target edits remains a concern. Another challenge is the complexity of gene interactions. Genes do not function in isolation, but rather as part of a complex and interconnected network. Altering one gene can have unforeseen ripple effects on other genes and biological pathways, leading to unintended consequences. Predicting these downstream effects is a major challenge in gene editing. Furthermore, the long-term effects of gene editing are still largely unknown. Changes made to the genome, especially germline edits, will be passed down to future generations. It is impossible to fully predict how these changes will manifest over time and whether they will have any unforeseen negative consequences for future generations. The potential for unintended consequences highlights the need for caution, rigorous testing, and ongoing monitoring of gene editing applications. Section 8. CRISPR for a spacefaring future. As humanity sets its sights on exploring and potentially colonizing other planets, CRISPR emerges as a tool that could play a crucial role in our extraterrestrial endeavors. The harsh and unforgiving conditions of space pose significant challenges to human health and survival. CRISPR could be used to enhance our ability to withstand these challenges and adapt to alien environments. One area of exploration is using CRISPR to enhance our resistance to radiation. Space is permeated with harmful radiation, posing a significant risk to astronauts on long-duration missions. CRISPR could be used to modify genes involved in DNA repair and radiation resistance, making humans more resilient to the damaging effects of cosmic rays. Another possibility is using CRISPR to tailor our physiology to the specific conditions of other planets. For example, CRISPR could be used to enhance our ability to utilize different energy sources, tolerate extreme temperatures, or resist the gravitational forces of other planets. This could involve modifying genes involved in metabolism, thermoregulation, or bone density. While these applications are still speculative, they highlight the potential of CRISPR to facilitate human adaptation to extraterrestrial environments. The prospect of using CRISPR to create a new breed of spacefaring humans raises ethical questions about our responsibility towards future generations and the potential for creating a genetic divide between Earth-bound humans and their spacefaring counterparts. Section 9. A future shaped by CRISPR. CRISPR has ushered in a new era of genetic engineering, one with the potential to reshape the world as we know it. From curing diseases to extending lifespan and even adapting humans for space travel, the possibilities seem endless. However, this power comes with great responsibility. As we venture further into the uncharted territory of gene editing, we must proceed with caution, guided by ethical considerations and a deep respect for the sanctity of life and the interconnectedness of all living things. The future shaped by CRISPR is not predetermined. It will be shaped by the choices we make today. Open and transparent dialogue, robust ethical frameworks, and strict regulation will be crucial in ensuring that this powerful technology is used responsibly for the benefit of all humankind. The story of CRISPR is still being written. It is a story of scientific ingenuity, human potential, and the profound responsibility that comes with wielding the power to rewrite the code of life.